So, in your drug experiments, and before we start going into what all of these experimental methods are, we're going to have a quick look at why we conduct experiments and a few of the key terms you're going to need to know when we go into analysing the strengths and limitations of these in our next lesson. So the first question is, why do we need to carry out experiments? And the real reason is we want to try and establish cause and effect. So do the changes in the independent variable bring about the results in our dependent variable? And as long as we control a lot of other variables, we can quite confidently state that this is the case. So the first thing we're going to have a look at now are the different types of validity. And internal validity refers to whether a procedure is actually measuring what it's supposed to measure. So are the changes in our results in the measurement of our dependent variable, are they down to the independent variable or not? Are they down to other factors or other extraneous variables? If the results are down to other extraneous variables, then the study lacks internal validity. If the results are going to be mainly down to our independent variable, then the study has high levels of internal validity. Whereas external validity, on the other hand, is all about can we generalize the results. So once we've got our findings, once we've made our conclusions, can we generalize our findings to other populations, other parts of the world, to other genders? Are we able to generalize the results of that study or not? If we can, brilliant, it has high levels of external validity. If we can't generalize our findings, then it's going to have low levels of external validity. So we're going to have a look at a few of the things that might affect internal and external validity in a little bit more detail. And internal validity can be reduced by many factors, such as demand characteristics. Remember, demand characteristics are if the participant manages to guess what the aims of the experiment are, they'll change their behaviour to try and give the experiment the results that they want or they think they're seeking. Investigator effects might also reduce internal validity. So this is where the researcher's expectations affect participant behaviour. It might be subtle clues or subtle changes in the way they word things or slight changes in their body language that affect the participant's behaviour. Situational variables can also have an effect. These are things like the time of day or how many hours sleep someone has had, which obviously can reduce the internal validity of a study. Order effects we've already discussed a lot in previous lessons about the effect of learning or boredom, and as long as we can control order effects, we can have high levels of internal validity. Other factors might include participant variables. So in certain studies regarding memory, concentration, there's participant variables such as IQ, which are going to have a clear effect on the results. People with high IQ are more likely to do well in a concentration task or in a memory task. So these are things we've got to control in our studies. And also social desirability. So social desirability is where people behave in a certain way because they want to portray themselves in a better light. So if I give you an example of someone doing a study on children at home, and they're asking mothers to keep a diary for 1 week about how their children behave at home. You'll find that the vast majority of mothers put down that their children are little angels and don't misbehave, whereas in fact they probably do. All small children will misbehave now and again. But the mothers in the experiment show social desirability bias. They don't want to come across as bad mothers. They want to come across as the best mother they can possibly be. So they put down that their child never misbehaves because that puts them across as a better parent. So social desirability is another factor that can reduce the internal validity of the study. So external validity can also be affected by certain things. For example, the place or the setting. So often studies that are done in labs will lack ecological validity because we can't generalize those findings beyond that particular environment. A laboratory is a very artificial environment, and therefore we can't always generalize the results to the wider world. You've also got population validity, which is if the study is all done on one particular group of people, such as males, or all done on Western population, such as the United Kingdom or the United States of America, then the study can lack population validity. It's all based on one population, and we can't necessarily generalize it to the wider world. You've also got temporal validity, and this is looking at when the study was actually done. So some studies will lack temporal validity if they were maybe done 50, 100 years ago. Because society's changed so much, attitudes, beliefs in society have changed, we can't necessarily generalize the findings from that time period to our time period. And finally, we can enhance external validity through replication. So if we can repeat the results and get the same ones over and over again, that does give us a high level of external validity. So before we get into the key points for all different types of studies, we're going to give you a very quick definition of each one. So a laboratory study is something that's carried out in a very controlled condition. A field study is carried out in a more natural environment. A natural study, which we're going to be looking at, is an experiment where the experimenter doesn't really control the independent variable. They're not directly manipulating it. It's something that occurs naturally. A quasi-experiment is not a true experiment. It's an almost experiment. And the IV is something that occurs naturally, such as gender. So it's not being manipulated, but it allows us to look at differences between different genders, for example. And finally, it's this idea of ecological validity. And this is the extent to which a situation can reflect real life. We're not going to be going through all the strengths and limitations. This is going to be something that we're going to be doing in class next lesson. But we are going to be looking at the key terms you'll want to be getting into an answer if they ever ask you about what is a lab study, field study, etc. So a laboratory experiment should be quite simple for you to imagine. It's going to be in a room. It's in a controlled environment. And by that we mean a lot of the situational variables can be controlled. We can control things like IQ, we can control the participants, we can control their age, we can control the time of day it's being done, we can control their diet if we want, we can control the amount of hours of sleep they've had, the amount of caffeine they've had, alcohol they've had. It can be a very controlled setting where we can control almost all variables. Obviously the participants are going to be aware that they are involved in the experiment, but they won't necessarily know the aims, and that's what we don't really want them to know. In most psychology experiments, if they know the aims, they start to change their behaviour. So we don't want them to know the aims if possible. And also in laboratory experiments, the participants are often randomly allocated to groups. So by this I mean it could just be as simple as pulling names out of a hat. It might be putting them into a number generator where it puts the participants into different groups and different conditions for you. And the whole idea of this is that it reduces any bias. It doesn't mean that if we were doing a study on the effects of memory on concentration, it doesn't mean we're going to get one group of people with all the high IQ and obviously going to have a bit of memory, and all the people with low IQ in another group. By randomly allocating to the groups, it allows us to reduce any bias, and it will be all completely random as to who is assigned to what group. So a field experiment is different to a lab experiment because it's done in a more natural environment. And however, the independent variable is still being manipulated by the experimenter. You are able to control some of the variables, and we're going to have a look at an example experiment in a minute. Some of you may have done it when you looked at conformity. But in most of the time, the participants do not know that they are taking part in the study, which is brilliant because it reduces any demand characteristics, any social desirability, because they're unaware they're actually taking part in one. So let's have a look at an example of a field study. We've got Hoffling's study, it's in the textbook. And what happens here is he had to make a phone call to a number of nurses. He phoned up 22 different nurses and he ordered them, his name was Dr. Smith, and he ordered the nurses to give a drug of Astrotin to some patients. And the dependent variable in this was whether they gave the drug to the patients or not. So estrogen isn't a harmful drug. It wouldn't have done any damage or had any long-term side effects to the patients. But this was all about, do nurses follow an authority figure? Hoffling rang up just saying he was a doctor. He didn't have any authority over them at all. He wasn't a qualified medical practitioner. He was simply a psychologist ringing up to see would they follow an instruction if it was given by a doctor. So he rang up, and what they found was that 21 out of 22 nurses did as they were told. So this is an example of a field experiment because it's not in a laboratory, it's in a natural setting. They're in a hospital at the time. However, Hoefling was able to change the independent variable. He was able to give that instruction in order to see whether it had an effect on their behaviour. So that's what makes this a field study. So a natural experiment, on the other hand, is always carried out in the field. And this is where an experimenter doesn't really manipulate any independent variable. They're not directly manipulating it in any way. And the independent variable is always naturally occurring. It might be the gender, or it might be privation, which is where you're kept away from your primary caregiver for a long period of time. We'll look a bit more of that when we do the attachment unit. But these sort of studies aren't really true experiments because an independent variable has to be manipulated in order for something to be regarded as a true experiment. So if we have a look at Charlton's example of what a natural study would look like. So in 2000, Charlton managed to study one small island in the Atlantic called St. Helena. Before 1995, there was no television there whatsoever. And his study was to look at the effects of Western TV on social behaviour. So as soon as TV got put into West Helena in 1995, they started to look at the behaviour. They looked at pro-social behaviour, so people acting nicer towards each other, and anti-social behaviour, people acting in a more aggressive way towards each other. And what they found was that there was no difference whatsoever in pro or anti-social behaviour as a result of Western TV being introduced. So the IV here, the independent variable, is the fact that we had no TV initially and later they were exposed to television. So there is an independent variable, but this was not controlled by the researchers. This was something that occurred naturally, it was happening anyway, and the researchers sort of took advantage of this naturally occurring situation. And it's something that they wouldn't really be able to do otherwise. So they were lucky that the TV was being put over there, and it gave them a chance to see whether TV has an effect on pro and antisocial behaviour. So that's what makes it a natural experiment. They had no control over what was happening, they just took advantage of an already occurring situation. So quasi-experiments are these almost experiments. Again, they're not a true experiment because the researcher isn't manipulating any independent variables. The independent variable in this case is always naturally occurring. However, the dependent variable could quite easily be measured in a lab. We'll give you an example of one in a minute. And the key thing with a quasi-experiment is that the independent variable is something that is naturally occurring. It's not being manipulated, it's not being controlled by any researchers or any experimenters. This is all something that's naturally occurring, like gender. So, for example, if we look at Sheridan's 1972 study, he wanted to test the obedience of giving electric shocks. These aren't life-threatening shocks, but electric shocks with increasing strength to a puppy. And what they found was that males, 54% of males went up to the maximum shock level. Again, like I said, it's not a fatal shock. They were just testing if people were given probes, would they go to the maximum voltage and give a shock to a small, tiny, defenseless puppy. So 54% of men did, and staggeringly, 100% of women went to maximum shock. So again, the independent variable here is gender. We're looking at men compared to women, and it hasn't been manipulated. This is something that's naturally occurring, and the researchers have just taken advantage of it, and that's what makes it a quasi-experiment.